My name is Peadar Lawlor. I work in the pig department in Teagasc in Moore Park, and I'm going to speak about the use of feed enzymes in finisher pig diets to improve production performance. With regard to the feed enzyme work, that work was conducted in Moore Park as part of the EcoFCE project, which is an EU-funded project. The people that would have worked on that, principally the PhD students, would have been Alberto Peadar and Ursula McCormack. And really what I'm going to present is work on results from 3 separate trials that were conducted as part of that project. One was where we fed enzymes in diets with alternative protein sources to soya, so difficult diets that contained wheat DDGS and rapeseed meal. So we added enzymes like phytase, xylanase and beta-glucanase, and we added protease to those diets. So the diets were formulated down in energy and amino acids, and we would have hoped to have seen an improvement in growth rate, possibly, and definitely in terms of feed efficiency. And I suppose the reality with that first trial was that the only benefit we saw from enzymes really was with regard to phytase. The phosphorus-sparing effect of phytase was proved in the study, but that was expected. We expected to see that. What we did find is that we had a response to protease in terms of feed efficiency. So feed efficiency was improved in male pigs where protease was fed, but we didn't see a response in the female pigs. And that may be because additional enzymes are required by males because they have a higher lean tissue growth rate, and because of that we saw the response from protease. The second trial we looked at was where we soaked a cereal component of a diet prior to feeding it, and the idea behind that was the soaking process would allow time for the enzyme to work in advance of feeding the diet to the pig, so that you get some pre-digestion almost of the diet. And in that experiment, our diets were cereal-based diets with barley wheat, pollard, soybean meal, and we added a beta-glucanase and beta-xylanase to the diet. In that particular experiment, we didn't see any response in terms of growth performance or feed efficiency in the pigs. We did see that when the animals were killed at the end or slaughtered, we saw that lean meat percentage was lower actually where we had fed the enzyme-based diets, and that was because the animals had become fatter prior to slaughter. And that probably indicates to us that there was additional energy released by the enzymes, but there wasn't sufficient amino acids there for the pigs to lay down additional lean tissue, so instead it was laid down as fat. The last trial we looked at, we fed, it was a bean diet. Again, we were looking at alternatives to high protein replacements for soya. We were interested in that as part of the study as well. So beans we thought would be a difficult diet to work with, so we said we'd add beans to the diet. They went into the diets at about 40%. First of all, the 2 enzymes we used were protease again, so that we were hoping to see some breakdown of protein into its amino acids and to get an improvement in that regard. Beta-galactosidase was added to the diet as well. So there was a number of aspects to that trial, but I suppose primarily to do with enzymes, the most important response we saw was that the protease was effective, and it was effective in improving feed conversion efficiency in the pigs on that trial. And what it did really was that it reduced intake on those pigs, and because intake was reduced and growth rate remained the same, we had an improvement in terms of feed efficiency from feeding the protease. So there is a benefit from using enzymes, but we have to be clever to match the enzyme, the correct enzyme, to the correct ingredient, and we have to formulate the diets correctly to be able to see the benefit from the enzymes.